So welcome everyone. We'll probably kick off now. We'll have a few more people joining the session in due course, but we'll just get started since we do have limited time. We've got 7 presentations today, which is really exciting. So welcome to the 5th and final special student session. It's on soil system science, and we have a special guest, Dr. Ngwanadasi Chiranda, who's joined us all the way from Colombia at the moment. He was meant to be in Morocco, living in Morocco by now, but obviously the world has had other plans for Ngoni in the meantime. So I'll just go through the structure of the session again. I think you're all quite familiar with them by now, but we will have a brief introduction by Ngoni to soil systems, greenhouse gas management science in general, and then we'll have the 7 student presentations, and then we'll have a discussion session at the end, which we strongly encourage the research supervisors and any other senior researchers on this call to participate in as well. That's one of the main reasons why we have the sessions. And yeah, definitely encourage engagement from student to student as well, since you're all here. So in saying that, please use the chat function in this session to record questions. We won't be doing the questions in the discussion until the end, so please do write down your questions as you listen to the presentations. And yeah, I think that's probably all you need to know for now. So yeah, I just want to introduce Ngoni, who's also a good friend of mine, and yeah, met him I think a year and a bit ago when I had first started working for the Secretariat. Was a very fresh face, and yeah, we got to spend time together in Colombia, which was fantastic at CART. And Ngoni was one of the kind of key founders of the CLIFF program, which was the precursor to the CLIFF-GRADS program. So Ngoni's actually been involved in the CLIFF-GRADS program since long before I have. So he's very experienced in this. He is a soil and climate change scientist with a scientific background in soil science and agroecology. He did postdoctorate research at Aarhus University in Denmark. I believe as well, Ngoni was lead author on the IPCC's 2019 refinement to the 2006 guidelines, which we discussed a bit with Olia Glade and also with Andreas Wilks. And Ngoni is also a co-lead to the GRA's Inventories and Nationally Determined Contributions Network, along with myself and 2 others from Spain and Australia. And I also happen to know that Ngoni is a podcast and audiobook fanatic, and that he listens to podcasts and audiobooks at 1.5 times speed, which I think is really efficient. I like to think I'm an efficient person, but I don't listen to podcasts and audiobooks at 1.5 times speed. So yeah, Ngoni, the floor is yours. Thank you for joining us today.
Thank you very much, Hazel. I'm really looking forward to this afternoon and from learning from the students on what they are going to be doing. So I will share my screen. Can you see my slides? Okay, great. Okay, so basically today we're going to be having a lot of presentations related to soil systems, the micro and the macro level. As you are aware, soil is it's quite heterogeneous, and it's really, I mean, if you look in the literature, there are different studies that show different and sometimes contradictory results. And one of the reasons for that is because of the heterogeneity and the complexity of soils. But then on top of that, agriculture, I usually like to say agriculture is a culture. I mean, it's the way you do things, right? The culture of agriculture aims to effectively use available resources, including the soil. And that means, and the objective of agriculture is to put food on our tables and money in our pockets. So how then do we make soils productive? So I've been thinking a lot about this, and one of the key things in order to make soils productive is management. So how productive soils are going to be depends on how you manage the soils. So management is very critical. So for change to happen, okay. So for change to happen, our research and the research that we're going to hear about today needs to inform management improvements at different scales. So there is management in terms of at the policy level, there is management at the regional level, there is management at the farm level, and that is the management that then affects soils. So basically all the policies and all the regulations end up incentivizing or decentivizing the way you manage your soils. So management is the key. I'll give you an example of some of the work that was done in Colombia. So if you look at that graph, you can see there that if you are moving from a degraded pasture where management is poor to a pasture that is some form of management, you have a grass legume pasture with fertilization, you can see there that your live weight gain is actually increasing about 4 times more just by management. So management is critical to move you from degraded, to move your productivity from 110 to around 450 live weight gain per hectare per year. So management is key. Of the work that we did with Siniro, who's also on this call, and some other students, CLIFF-GRADS students, was that they did, we looked at what management would do to emissions of nitrous oxide from a pasture that is degraded or a pasture that is managed. So what we observed there is that you see the dots in black is where there is low vegetative cover. So the management is poor. And you can see there that the emissions of nitrous oxide from the urine deposited on these degraded pastures is actually higher all the time compared to where there is some management. So management is critical in reducing your emissions. So it's critical in increasing your productivity. It's also critical in reducing your greenhouse gas emissions. My supervisor once told me that, Ngoni, if whatever you are doing does not put food on the farmer's table or money in their pocket, you can as well forget it. So in some way, all our research needs to be contributing. The outcome of our research should be food on tables and money in pockets. Maybe today, as we listen to the different presentations from the different students, let's view these presentations from the lens of, are we putting food on the table and money in the pocket? And the key to doing this is how we manage the soil. So management is critical for us to achieve this. So hopefully, as we listen to all the talks today, we are going to see the importance of management. Thanks, Hazel.
Thanks so much, Ngoni. Yeah, that absolutely is the key. If something is going to affect the profitability of farming, then obviously it's not essentially a solution because it's not going to be taken up. So great, thank you. I'm just wondering if there are any questions for Ngoni before we move on to the student presentations. Please feel free to unmute yourself and ask a question if you do have one.
Hi, Ngoni. It's Florencia from Argentina. I was curious about the difference between management or not. In the 3 points that you show in the beginning, there wasn't huge difference, but it was in Brazil, and I don't remember the other point. What will you attribute that difference to?
Thanks, Florencia. Let me go back to the... Okay, so this is Colombia. So it seems like there was no difference. So when you zoom in, so if you look at this graph here, can you? Am I sharing my screen?
Not at the moment.
Not at the moment. So. Okay. So am I sharing now?
Yes.
Okay. So if you look here, and I think this is Soluma in Colombia. When you zoom in, because it's basically because of the scale, you cannot see that. It seems as if there is no difference. But when you zoom in, you can see there that there is a difference between the degraded and the non-degraded pasture. So in some cases, I mean, like in Argentina, I think the challenge was that I think it was cold. So when you have the emissions were lower in both the degraded and the non-degraded, hence the low emission factor. But generally, if you zoomed in, you could see that even under those conditions, management was still making a difference. So there is an issue of the heterogeneity of the soil, but as you can clearly see there, even under those conditions, the management is what is making a difference.
Thanks,
Hello, Ngoni. Hello, Ngoni.
Hi, hello, hi.
Yeah, this is Abubakar, your mentee.
Oh, okay, hi, Abubakar. Yeah, sure. How are you?
Yeah, I'm fine. Fine. Yeah, going by your presentations, more food in the table and more money in the farmer's pocket. What can you say about practices involving reduction in emissions? Because there is a lot of controversies, like on the management practices. When you are targeting low emissions, it is likely you are going to have low productivity, maybe in terms of reducing water quality, as some water use efficiency. Some literatures are reporting maybe reduction in yield with the adoption of alternate wetting and drying management practices over the continuous flooding. There is a lot of controversies regarding that. When you are targeting low emission, is going to have sometimes negative effects on the productivity. When you are targeting high productivity, sometimes you are going to have high emission factors. So what can you say about this? Thanks.
Okay, thank you very much. That's a very good question. I think if you look at it, I mean, let's step back a little bit. Where are emissions coming from? Really, emissions are a product of inefficiency. Whenever you see emissions in any system, it basically means you are using something in an inefficient way. So if, for instance, you have water all the time, I think Ole talked about alternate wetting and drying before, right? If you have continuous flooding, you are using water inefficiently, because even though rice is a thirsty crop, it's not thirsty all the time. So when you use something in your system inefficiently, you end up getting emissions. So for instance, if you have a lot of fertilizer, that means you are using the fertilizer in an inefficient way, then you have a lot of nitrogen available for loss, right? So emissions are a source of inefficiency in the system. Having said that, I think it now is on you and me, I mean, the researchers, the new generation, to see how can we then improve through management the efficiency of our systems. If we improve the efficiency of our systems, we, as a byproduct, we also reduce the emissions that are coming from those systems. But of course, in some cases, I mean, the challenge is you need regulations, you need policies in order to curb the greed, or to curb because the moment you produce more, you want to expand, right? And generally, farmers, when they have more production, like for instance, in the case of the discussion in the livestock sector, where if you intensify, farmers produce more, then they end up clearing more land to produce more. So you need policy to then regulate the kind of greed of human greed, no? So I think the short answer is, it is on you and me to conduct research to improve efficiency. and that will only be done through management. So we still get back to the saying, management is the key to both increasing productivity and reduce greenhouse gas emissions. Sorry for a long winding.
Great, thank you, Ngoni. So we have one more question from Alex. We'll have time for your question, Alex, if you would like to unmute yourself, and then we'll move on to the student presentations.
Yep, yes. Thank you. Can you hear me?
Yes.
Yes, you can hear me?
Yes.
Yes, Alex, we can hear you.
Yeah. Yes, my question is about population and management. In the slide you have shown, for example, about the countries, is there a relationship between management and population in terms of the emission factor versus productivity? That's my question.
Okay, thank you. Okay, sorry, I did not. I think I didn't explain mostly what we did there. So what we basically did is that we took urine from cattle, and we deposited this urine in a degraded pasture and a managed pasture. And what we see there is that every time the pasture is degraded, the emissions were higher, the nitrous oxide emissions were higher in that pasture compared to where you had management. So the idea is then, of course, if you then increase your population of animals, that might also switch that, right? Because maybe in the well-managed pasture, you can have more cattle, right? So you might end up even having more emissions. But the challenge is the other benefits to having a well-managed pasture. The first is you are increasing the productivity of your animals, which is the objective of farming, right? More food on the table and more money in the pocket. You also have more root biomass. So if your pasture is well managed, you have more roots that are from the pastures that you'll be putting into the soil. So if you account for that as well, you might actually end up having a net negative emission. So, but then you need to account for all these different other aspects. And the degraded pasture, basically, you are not producing so much. You are also not having so much carbon into the system, and you also have a lot of emissions from urine that is deposited. So if you then have a high population of cattle, of course, you probably won't have so high because there won't be so much feed. But if in the sense that if you have a lot of cattle there, you would have a lot of emissions, no sequestration and no production. So that is the way it links up with cattle population. Thank you.
Great, thanks, Ngoni.
Okay. Thank you.
Yeah, another excellent answer. I think we will move on to the student presentations now. So Maria, it would be great to start with you since we've already checked your audio, if you are able to unmute and share your screen.
Yep. Okay. Can you hear me well?
And see you as well.
Okay.
We can hear you, Maria.
Okay. Is it fine?
Yes, your screen is great. Thank you. Please go ahead.
Okay. So, hello, my name is Maria Elisa. I am PhD candidate from Federal University of Paraná in Brazil. And now I am guest researcher at ISRIC World Soil Information here in Wageningen in the Netherlands. My PhD project is called Agrohydrological Modeling and Soil Mapping in Brazil, and I am going to show you a little bit of it. Okay, let me pass this slide. Okay, so my project is in a context, something between soil mapping and soil physics. We are basically concerned about soil hydraulic parameters such as the soil water retention and the soil hydraulic conductivity because they are major keys for hydrological modeling. And the objective would be something related to development of methodologies to generate georeferenced agrohydraulic estimation for Brazilian soils using process-based models and available soil data. the basic idea is to look for patterns. And we do that combining soil maps and PTFs and somehow evaluating the uncertainty and the results of it. The first stage of my project I did when I was a guest researcher at the University of São Paulo in Brazil. I was there for 1.5 years. Basically, we study the field capacity in Brazilian soils, producing soil maps based on data available on soil grids, which is a product from ISRIC, where I work now. We combined that with PTFs, and we used the swap model to simulate a drainage experiment and see the results for field capacity under different criteria. And then we try to evaluate the uncertainty related to that, which is a major key in this kind of analysis, because we have uncertainty related to the soil maps, to the model approach, to the measurements, to the PTFs that we use for the soil hydraulic conductivity, but also for the PTFs that were used for soil water content. It also depends on the flux we use and also the scale. But we could observe some interesting things, such as the field capacity values were lower than they usually adopted for temperate soils, which also corroborate with another measurement experiments. But one major key, which is the uncertainty, is kind of related with what I am doing here at ISRIC. Here I am going to work basically in 2 projects. So I started in September, and I'm going to stay until March of the next year. One of the projects is mapping soil organic carbon change to support climate change mitigations, which is basically mapping soil organic stocks in space and time. I still didn't start to work with that, but they developed a first part of this project, is already published, and the idea is to extend it globally. The first part was made just for Argentina. What I've been doing here in this almost 2 months, I'm working in mapping derived soil hydrological properties using machine learning. So we have the WOSIS database, which is a collection of data from the whole world in a standardized way. And we are interested in the water retention at 100, 330, and 1500 centimeters. So basically we have to convert the data from gravimetric to volumetric when that's the case. We have to check the consistency of that. And after we have like a organized database, we want to apply digital soil mapping techniques, test covariates, and use this model to predict the points that we don't have measured. And within all these mapping techniques, one of the important things is the evaluation of uncertainty. And that's somehow, that's the major point that I want to link with my PhD project. So that's it in my 5 minutes. Thank you very much for listening. I am open to questions in the last minutes.
Great, that was awesome. Thank you very much, Maria. I have some questions for you, but we'll ask them in the discussion session at the end. So for now, I will like to hand the floor to Eric Rodrigo da Silva Santos from Brazil also, who will be studying at Bangor University. So Eric, if you are there and able to unmute yourself and share your screen. Perfect.
I'm just trying to put in the presentation mode. Can you see the presentation mode?
It looks great.
Yeah, it's in the presentation. All right. Yes, just Hello, everyone. I'm Eric Santos, and I'm from Brazil, as Hazel said. And then currently, I'm doing my research stay at Bangor University with Dr. David Jones. And back in Florida, where I'm studying at the University of Florida, I'm doing a project about grazing cover crops, and we are studying alternatives to improve sustainability and row crop productivity in North Florida. And the Florida scenario that we have there is that a lot of the lands, most of the producers, they plant like row crops, for example, cotton and peanuts. And this land, most of this land also is under irrigation. And whenever the warm season is over, they do nothing with the land. My presentation is busy, sorry about that. They do nothing with the land. And we can see a scenario as this one we have in here in the cool season scenario, where they do not plant even cover crops in there. And a problem that we have in the region specifically that I'm doing my study is that Jackson County also has the Florida Blue Springs, which is a first magnitude springs. And there's an increasing level of nitrates in the last decades, as we can see here in this figure, from 1958 to around 2000 here. We can see the concentration of nitrates in the water in the Blue Springs, and it's crazy. It's increasing a lot. So basically, a cover crop is any crop that can grow to provide soil cover, regardless if it's going to be incorporated later in the soil or not. And in this context, we have some really good cool season forages that we can use as cover crops. For example, ryegrass, oats, and triticale. And whenever we have animals grazing these forages, they can improve their performance as well compared to whenever they're grazing warm season forages. So, but there's a lot of space between what's grazing, right? As we were discussing earlier, everything's about management, and we don't want to like overgrazing the forage, and we don't want undergrazing the forage because we're gonna be losing per area, or we're gonna be losing animal gain per animal, right? So we want to find some optimal zone between these two types of grazing management. And with that, I'm testing 9 different systems there. First one, the first set of treatments, I call them control, which is basically a conventional row crop. In the first treatment, we have only row crops during the summer and no cover during the winter. In the second treatment, we have crops during the summer, which is cotton, and then we have cover crops during the winter at 34, and we apply 34 kilograms of nitrogen. And the third one, we have the same, but during the winter, we apply 90 kilograms of nitrogen. And then we have the intercrop livestock, which we have the same as the treatment tree, but we put the animals to graze the cover crops during the winter. For example, here in this treatment, the animals, they graze the cover crops every 2 weeks, and they leave a residue of 2,500 kilograms of dry matter per hectare. And in the second, they leave 15, and in the third system, they leave 500 kilograms of dry matter per hectare after the grazing. And these treatments are to be undergrazed, moderate graze, and overgrazed treatments. And we have a similar set of treatments here. But instead of row crops during the summer, they have warm season grasses. And the animals, they're gonna graze these forages as the same way as they did in the set of treatments before, with undergrazing, moderate grazing, and overgrazing. And some of the ongoing experiments that we have right now are we are evaluating the cover crops above and below ground litter decomposition. And in the second study, we are evaluating the soil chemical and physical properties. as affected by graze intensity. And in the third experiment, we are evaluating the nitrate leaching and row crop productivity as affected by cover crop and grazing management. So here in the experiment 1, we are estimating the litter decomposition rate and the crop nutrient uptake according to the graze intensity and rotational systems. And here are some variables that we are measuring as litter deposition, biomass, and nitrogen disappearance. And basically, we put some income bags in the soil. And after a period of time, for example, 4 days, 8 days, 16 days, 32 days, until 128 days, we go there and we collect these litter bags and we analyze for carbon, nitrogen, and as the detergent and insoluble nitrogen as well. And then in the second study, we collect soils from 0 to 15 centimeters and also from 0 to 90 centimeters. And we separate the soils in different fractions and we analyze for soil particulate organic matter fertility and also for ammonia and nitrate mobilization in the soil, for example. And here are some of the pictures that I have from this study, where we collect the soils at 90 centimeters depth. And also, as you can see, like the POM here and MAOM is like the particulate organic matter, which is mixed with some sand here. And here we have the mineral associated organic matter, but this is only from the first 15 centimeters layer. And in the third experiment, we evaluated the nitrate leach and row crop productivity as affected by cover crop grazing management. And here we evaluated all the pasture responses as herbage mass and herbage accumulation and digestibility and crude protein. And we also evaluated the cotton yield at the end of the season and the nitrate leaching in the water. As we can see, we have these big lysimeters in every single plot, and they are about like 3 meters deep. And that's and we go there every 2 weeks and we collect our water samples. It is everything that I have for today. I'm working the data analysis. I have some of the results, but it would be too long. But I'm glad to share with you if you email me. Thank you.
Thanks so much, Eric. That was really great. I'm just wondering if we can go to either Anthony or Ricardo. I see Anthony's joining and then dropping off. Perhaps, Anthony, we can try to see if you can unmute. Share your screen.
Hello, everyone.
Hi, Anthony. Great. We can hear you. So are you able to share your screen as well?
Hello, I won't be able to share my screen because I'm using my mobile phone. My network is very poor at this side, so I'm connected over the phone.
Okay, right. Okay, in that case, we'll go to Ricardo because I don't have your presentation open yet. So we'll open your presentation and we'll let Ricardo do his presentation now, and then we can go from there. So I'll just mute you again, Anthony, and if you're here, Ricardo, if you can please share your screen.
Okay, that's fine.
Are you there, Ricardo? We can't hear you at the moment, Ricardo.
Hello.
Hello.
Can you? I cannot share my screen.
You can't share your screen either. Okay. In that case, we'll start with one of the presentations that needs to be presented online, and we'll prepare your 2 presentations, and then we will go from there. So I will start with Amanoi. Amanoi George Aminchwe, who is from Cameroon. He is being hosted by the USDA Agricultural Research Service, the Northern Plains Research Lab in Sydney in Montana. And his PhD is the role of beneficial management practices for reducing soil greenhouse gas emissions and enhancing soil carbon sequestration from maize production systems in Cameroon. So I will share my screen now. Can you please make sure that everyone else is on mute?
Hi everyone, my name is Emmanuel George Amentre. I am from Cameroon. My CLIFF-GRADS host institution is the Northern Plains Agricultural Research Laboratory in Sydney, Montana, USA. In my PhD thesis back here in Cameroon, I am working on the role of beneficial management practices on reducing soil greenhouse gas emissions and ensuring soil carbon sequestration in maize production systems in Boya, Cameroon. Agriculture emits between 5.0 to 5.8 gigatons equivalent of carbon dioxide in a year, and this is up to 13% of the total anthropogenic greenhouse gases being emitted into the atmosphere by humans globally. Even though Africa's agriculture local tools and methods, emissions from the agricultural sector in Africa are among the fastest growing emissions in the world. Within the cropland systems, the tillage regime being adopted and the soil amendments being added into the soil to boost productivity has an impact on soil greenhouse gas emissions and soil carbon sequestration. In the face of climate change and many other environmental challenges, the continent's biggest problem is to find a soil management practice that generates less greenhouse gas emissions and ensures more carbon sequestration, and at the same time ensuring food security. The first objective of the research is to measure the cumulative growing season greenhouse gas emissions as affected by tillage and crop amendments. The second objective is to measure the changes in soil microbial biomass, carbon, and nitrogen throughout the growing season as affected by tillage and soil amendments still. And the last objective is to examine the impact of tillage and crop amendment on maize use. This research will be carried out in 2 growing seasons. And to achieve this objective, a split plot design with 3 replications have been adopted at the University of Boya campus. To collect data on greenhouse gas emissions, we have installed flux density chambers where we manually extract gas with a syringe stored in airtight containers, and these gases are being sent to the Northern Plains laboratory for analysis with the gas chromatography. During these sampling events, we are also collecting data on soil water content, atmospheric air pressure, soil nitrate and ammonium, soil temperature and atmospheric temperature, because these are parameters that greatly have an impact on soil greenhouse gas emissions in agroecosystems. To collect data on soil microbial biomass, carbon, and nitrogen as affected by tillage and crop amendments, we will collect soil samples when the crop is 3 weeks, 6 weeks, 9 weeks, and 12 weeks. And these samples will be sent to the University of Chang laboratory, and the chloroform formication method will be used to measure soil microbial biomass, carbon, and nitrogen. When you must have gotten these results, we will use the analysis of variance to test if there's a significant difference between soil microbial biomass, carbon and nitrogen as affected by tillage and crop amendments. For the last objective, which is to measure maize use as affected by tillage and crop amendments, we will manually trash maize from their cobs when they are ready, measure the weight in kilograms and convert it in tons per hectare since the dimensions of the plots are already known. The study is being carried out at the University of Boya, which is found in the city of Boya, southwest region of Cameroon. Boya is located in the tropics in the slopes, along the slopes of Mount Cameroon, which is the second highest peak in Africa. Climate change is an emerging major threat to Africa's agriculture. And so this research will document the true contribution of soil management practices in reducing soil greenhouse gas emission and enhancing soil carbon sequestration in maize farms in Cameroon. Thank you very much for listening. I look forward to engaging with you in other sessions.
Great. So I think everyone can hear me again. Ricardo, we have loaded up your presentation. So if you can unmute yourself now, you can speak. So bear with me, I'll just introduce you. So we have Ricardo from Brazil, and he is studying at Massey University in New Zealand. Ricardo, if you could just let us know when you want us to go to the next slide, that would be great. Thank you. You requested to share your screen. Let's see. Are you there, Ricardo? We can't hear you at the moment.
Oh, sorry.
Yes, hello.
Hello. Can you allow me to share my screen?
Didn't realize you wanted to do that. Bear with me. You should have permission to share your screen now.
No, it's not possible.
Maybe it might take a second, but you could try now.
No.
I think you've joined twice, Ricardo, so perhaps.
Okay.
Can you share now? I've just given your other.
Okay.
Otherwise, we can share. There we go. Perfect. Great. All right, the floor is yours, Ricardo. Please go ahead.
Hello, my name is Ricardo Cesare. I'm from Brazil. My CLIFF-GRADS organization is School of Agriculture and Environment, Massey University, New Zealand. My PD title is Inoculation of Brachiaria with the plant growth promotion Rhizobacteria, Azospirillum and Nitrospirillum, impacting the biomass production and greenhouse gas emission. Brazil has a pastoral land area of 170 million hectares, and the majority of these pastures are planted with general brachiaria. And 50% of these pastures are in different states of degradation. And various factors contribute to pastoral degradation, including improper management and the others. And the management of pastoral products becomes a great challenge to the sustainable management of tropical environment and inoculation of different forest species with the plant growth promotion rhizobacteria, Azospirillum brasilensis, and Nitrospirillum can be an important ally in the sustainable management. Research objectives and select the plant growth promotion bacteria with the potential to consume nitric oxide in the soil, to evaluate the nitric oxide emission and biomass accumulation in brachiaria inoculated with Azospirillum brasilensis, to determine nitric oxide emission factor in pastoral areas of brachiaria humidicula inoculated with strains of Azospirillum. Method, experiment 1, nitric oxide emission from agricultural soil. Effect of soil inoculation with Nitrospirillum and Azospirillum brasilensis strain. In this experiment, 50 Azospirillum and 5 Nitrospirillum strains were used. Nitric oxide production in vitro and height between nitrogen and nitric oxide after for 24 hours. Experiment 2, plant growth promotion rhizobacteria increase the efficiency of fertilizer while reducing nitric oxide emission. And greenhouse gases pot experiment was conducted, were used 2 strains of Azospirillum and 2 species of brachiaria were inoculated, Brachiaria humidicula and Brachiaria decumbens. And nitric oxide flux was measured 22 times during 65 days after any application. And experiment 3, use Brachiaria humidicula inoculated with the plant growth promotion bacteria, Azospirillum brasilensis, reducing nitric oxide emission from cattle parts in the tropics. The third experiment will be conducted, 2 strains will be used, and 2 Brachiaria humidicula and Brachiaria resiliensis will be inoculated. Preliminary results. Experiment 1, nitric oxide emission. Nitric oxide emission per pot 0, 8, 6, and 20 hour after any application. And experiment 2, biomass production in Brachiaria humidicula and Marandu inoculated with the two strains of Azospirillum. Expected results. The mitigation potential of plant growth promotion rhizobacteria inoculated in the tropical pasture, improve the biomass production in Brazilian pasture, and to develop a strategy that improve the quality of pasture in stage of degradation. Thanks.
That was great. Thank you, Ricardo. We will move on to the next presentation.
Hello, everyone. My name is Anthony Mlambo, a PhD student with the Bindura University of Science and Education in Zimbabwe, and I'm a Zimbabwean. The title of my research is to assess the impacts of condor-based water harvesting technologies, soil water retention membranes, and nutrient management options on soil organocarbon accumulation and greenhouse gas emissions from coarse-textured soils in Zimbabwe. I have 3 objectives. The first objective is to determine the effects of soil water retention membranes, tide and standard contours on soil organic carbon accumulation in agriculture production. And paper 2 will be to measure and quantify carbon dioxide, methane and nitrous oxide from tide and standard contours, subsurface soil water retention membranes from coarse texture soils in Zimbabwe. And finally, paper 3 will be to determine the effects of different nutrient soil management practices on greenhouse gas emissions released from condor-based water harvesting technologies in Zimbabwe. Soil samples as a baseline from block 1, and these were collected from 0 to 10 centimeter depth, 10 to 20 centimeter, up to 80 to 100 centimeter depth. And the results indicated that organic carbon is relatively higher in the upper soil surface and decreases as we go down the soil profile. These results were almost similar in the second plot, where we also found that percent organic carbon is relatively higher in the upper layer of the soil surface and decreases as we go down the soil profile. We also did some soil temperature measurements from the two plots, the soil water retention treated plots and non-soil water retention treated plots. And in replication 1, week 1, the results indicates that soil water retention treated plots have relatively higher temperatures of about 37.38 degrees Celsius against 30.3 for non-treated soils. And overall, soil-treated soils with the soil water retention technologies have higher soil temperature as compared to non-treated soils. The results are also similar in replication 2, where soil water retention treated soils recorded a relatively higher soil temperature as compared to non-treated soils, as indicated by the bars in this slide. The research is being conducted in Ntare district in Marange, where 2 sites has been selected at Maocha and Marange homesteads, where soil water retention technologies and standard contours were established. Measurements of greenhouse gases will be done using static chambers beginning this rain season. Time series will be used to calculate greenhouse gas fluctuations, and also linear regression will be used to determine the relationship between soil water retention practices and carbon dioxide fluxes. In conclusion, so far the preliminary results have indicated that soil water retention technology increases soil temperature at 15 to 30 centimeter depth, and the amount of soil organic carbon is relatively higher in this upper soil layer as shown in the baseline results. Another sample of results after installing the soil water retention technology has been collected and submitted to Soil Chemistry Research Institute for analysis, and the results are still to be out from the lab. I thank you so much.
So I'm just going to shift now to Bethel Giramu, I apologise, Bethel, if I pronounced your name wrong. She's not able to actually join us today, but she is doing her PhD CLIFF-GRADS experience at the Soil and Water Conservation Research Unit in Pendleton in the USA. So I will put her presentation on now.
Hi everyone, I'm Bethel Garamo from Ethiopia, a PhD candidate in climate smart agriculture and biodiversity conservation at Haramaya University, Ethiopia. And before I start my presentation, I would like to thank the CLIFF-GRADS and my host organization, as well as my supervisor, Dr. Hero Gorani. So to start my presentation, let me introduce you the title for my PhD dissertation. It says Modeling Impact of Climate, Land Use, Land Cover Change and Soil Management on Soil Organic Carbon Stock using Century and Sequester Carbon Balance Model in Anjani Watershed, which is found in Amhara Regional State, Ethiopia. To give you some general introduction or general overview, as you know, the influence of human beings on the natural environment has been increasing or has been recognized since the Industrial Revolution. For example, the level of carbon dioxide, which is major greenhouse gas and inert as well as trace gas, has been increasing from since the Industrial Revolution, as well as agricultural productivity is becoming low. That leads to the changing climate and land use, land cover, and the increasing in population as well as the increasing in rapid infrastructure development are also expected to increase on ecosystem for food production. Besides to these changes or increases, to a rapid rise in release of carbon dioxide into the atmosphere and declining in soil carbon sequestration potential. As you know, soil is the largest terrestrial carbon pool, and this largest terrestrial carbon pool has been depleted due to land use, land cover change and climate change. And one of the key questions in most of soil organic carbon researchers are related to the fate of soil organic matter with changing in temperature and changing in land use, land cover change. So to solve this kind of research questions, there are different process-based models and empirical models which can be used and has been used in different countries. What I motivated to do my PhD dissertation in this area or in this field is understanding the dynamics of soil organic carbon can guide in establishing or management approach, different management approach for sustainable soil utilization, as well as in my country, Ethiopia. Several studies showed that the high variability in topography, the high variability in geology, organic material and climate have resulted in soil type and variability in Ethiopia. On the other hand, the massive land rehabilitation and conservation response by the government as well as different donor partners have been carried out since 1970s in Ethiopian calendar, but unfortunately past research in Ethiopia focused on soil organic matter or carbon from a plant nutrition point of view, while studies on carbon sequestration started only recently. Beside to this most previous research were more in soil degradation than rehabilitation or restoration aspects, and this studies has also not paid attention to use soil carbon models. So I have different general 4 independent experiments for my PhD. The first experiment is evaluation of Century and sequester carbon balance models and predict soil organic carbon stock under different land use in Anjani watershed. In this experiment, different soil physical and chemical properties will be analyzed based on the model procedure. Century and sequester models, initialization, validation, and calibration also will be performed. The second independent experiment is prediction of impact of climate change on soil organic carbon stock in Anjani watershed. In this experiment, I will also analyze soil organic carbon change from the period 2010 up to 2100 under 4 climate change scenarios and 1 baseline scenario. the 4 climate change scenarios will be obtained from coupled atmosphere ocean global climate models. And the third experiment will be assessment of soil organic carbon change during the period 1984 up to 2090 under different land use in Untitled watershed. And for this experiment, Landsat images of different bands will be downloaded and analyzed by using ArcGIS, and Landsat 5 terrain mapping and Landsat 7 ETM plus will be downloaded from the USGS, and them also generated from USGS. The final experiment, which is experiment 4, will be evaluation of different selected land and soil management practice on carbon sequestration and develop management recommendation in Untitled watershed. Rather than focusing only the challenges or the impacts, I will also evaluate different selected land and soil management practice on carbon sequestration potential, and I will also develop management recommendation for the future. I have also expected outcomes. The first one is the most widely used process-based soil carbon model, which is Century and Sequester, will be checked in the study area. Projected future regional scenarios of temperature and rainfall will be established in the watershed based on different combination of general circulation models and regional climate models, as well as regional climate projections for the near terms and mid-century periods. Different land use, land cover change maps will be also generated during the period of 1984 up to 2090, and the other expected output will be the mainland soil management practice that affects soil organic carbon pool or sequestration potential will be also identified. The base land soil management practice that will increase carbon sequestration and agricultural productivity will be suggested and implemented in the watershed. And finally, at least 2 research outputs will be also published. Thank you for your attention, and with this I will finalize my presentation. Thank you so much.
Great. So with that, we will shift to our 7th and final speaker, which is Chukwuebuka Christopher Okolo from Nigeria, and he will also be at Bangor University. So I will just bring up his presentation now.
My name is Chukwuebuka Christopher Okolo. I'm a Nigerian citizen, and my host institute is Bangor University, UK. My PhD title is Soil Organic Carbon and Total Nitrogen Sequestration, Microbial Biomass, Primary Effects, and Stability Indices on Different Land Use Types in Semi-Arid Ethiopia. The general overview of my study is all about improving soil organic carbon sequestration potential of dry land soils in semi-arid areas of northern Ethiopia, while the specific objectives include to assess and compare the impact of land use types on CO2 fluxes, microbial biomass, and soil biochemical properties in contrasting land use types, using different proxies of microbial activities and supporting sea labeling in semi-arid of northern Ethiopia. 2, to assess how variations in carbon and microbial biomass availability influence priming rate and magnitude. 3, to measure and compare soil organic carbon and total nitrogen concentrations and stocks of soils in contrasting land use types. And 4, to determine the magnitude of soil organic carbon concentration associated with various soil aggregate sizes using different approaches of dry sieving and wet sieving under different land use types. The summary of method of analysis: in general, 4 research activities regarding our research papers were conducted using standard laboratory analysis. So for research paper 1 and 2, the objectives focused on analyzing soil organic carbon and total nitrogen, microbial biomass carbon and nitrogen, water extractable organic carbon, amount of added glucose mineralized to 14 CO2, and amount of 14 CE microbial biomass carbon. Then in research paper 3, soil organic carbon, total nitrogen, cation exchange, bulk density, and particle size distribution were analyzed and studied at 3 depths of 0 to 30, 30 to 60 and 60 to 90, and the same depth the compartment is equally applicable to research paper 1 and 2. Then in research paper 4, I based my analysis on wet aggregate separation and dry aggregate separation, and try to use this data set to generate different aggregate indices. And I equally analyzed the content of soil organic carbon in different soil aggregates of micro and macro sizes. So this research was conducted in 4 different locations in semi-arid region or Tigray region of northern Ethiopia, namely in the Desa Forest, Hugumbuda Forest, Hakehalet watershed, and Geregera watershed. So for the Desa Forest and Hugumbuda Forest, they have 3 common land use types of forest, grazing land, and cropland, while in Hakehalet watershed and Geregera watershed, we have the exclosure, grazing land, and cropland. The key findings from this study: 1. Irrespective of land use type, microbial biomass carbon decreased with increasing depth as the emission of 14 CO2 and 14 C incorporation into microbial biomass. This indicates that microbial activity decreases with depth independent of land use type, but with few exceptions. Secondly, higher priming in natural ecosystem compared to cropland is an indication that conversion of natural ecosystem to continuous cropping not only will lead to depletion of primable pool of carbon, but also to changes in biogeochemical carbon cycling, as it led to an altered response of soil microbes to carbon inputs. Third, conversion of forest to cropland accounted for significant losses of soil organic carbon and total nitrogen, with considerable amount of CO2 emission to the atmosphere, while exclosure establishment supported restoration of degraded grazing lands with recovery of soil organic carbon and total nitrogen stocks, especially in the topsoil layer, though the duration of establishment and site-specific characteristics affect the pattern and magnitude of these dynamics. Then fourth, soil organic carbon has strong and weak positive correlation with structural stability index and aggregate stability index respectively, implying that soil organic carbon has stronger influence on soil structural stability index than aggregate stability index. Then in conclusion, establishment of exclosure as a land restorative mechanism should be continued, considering the huge impact in terms of offsetting carbon loss, especially in this current study where we have 10-year-old exclosure, and improving soil organic carbon pool in addition to enhancing ecosystem services. This study supported earlier reports that conversion of native forests to other land uses results in a significant decrease in the soil organic carbon stocks, microbial biomass, and increased CO2 emission to the atmosphere, thus contributing to global warming. Finally, dry sieving could as well serve as a quick, sustainable alternative to the more time-consuming and tedious wet sieving method in a semi-arid dry land in determining soil aggregate stability. Thanks.
Excellent. So that's all of our PhD students to speak. I'm wondering, Ngoni, if you're there, if you have any comments on any of the presentations that we've seen.
Okay, thanks a lot. Yes, I have a few comments, and maybe I'll start with the first presentation.
Ngoni,
Maria, right?
it's quite difficult to hear you. It's a little bit muffled.
All right, can you hear me now? Is it better?
That is better, yeah.
Okay, so I have a few questions, but I'll just go one by one. And maybe the first one will be to Maria. In terms of the data that you are collecting, are you facing any challenges with regards to data quality? Because I mean, one of the things is sometimes you have different laboratories using different approaches of analysis. Are you facing that kind of challenge in your work?
Yeah. Yes, I think that would be one of the major challenges in soil studies, especially in global level. We have, for example, for the work I am doing here at ISRIC, we have a really different distribution of the data. So we have many points at the USA, for example, but not much in Asia, not much in Brazil. So we have large areas without much data. And also we have to face, for example, we are working with soil water content, and sometimes it's measured gravimetrically, and we are not interested on that, so we have to convert it. It's kind of direct, but it has some errors. It carries some errors. So yes, data collecting is always a problem, especially globally, I would say.
Yeah, I think that, I mean, that's one of the challenges that you probably need to watch a lot, because I remember spending 3 months of my life working on some modeling, but then the challenge was the data was in the wrong format. So 3 months of my life, I was just working with the wrong data and getting crazy. And then when I just then asked the person who collected the data, I got the answer, and then everything was solved. So it's important for you to just take note of that.
Yeah, that's really important. I am trying to do that consistent analysis now. I was thinking it was something I could do like in 1 or 2 weeks, but it took me already 1 month, and I am still trying to get another approach and then see if it makes sense. Do not discard data that it's reasonable, but also do not use values that are clearly not realistic. So yeah, that's an important part.
While you're there, Maria, I'm wondering what the end use of a global soil carbon map will be. Would it largely be for people who are compiling inventories or for other uses?
Actually, I think I cannot answer this question now, because I really am not familiar with this project. But as far as I know, it's a global soil mapping of the carbon of the organic carbon stock, and they are evaluating spatial and temporal variation. Mm-hmm. So it's quite a big challenge, and probably in a very small scale, for example, less than 1 kilometer. So probably it's available within the soil grids variables, but I am just guessing, actually.
Sure. I wonder if Gerard is still on the line. No. And Ngoni, I will hand back to you if you had any other comments to make.
Yeah, no, just on that, I mean, just in terms of utility of such a map, I guess maybe, I don't know, but I'm just thinking that maybe you could use it to identify hotspots, right? Or places where you have degradation, or where you're losing carbon or something like that. So I'm just thinking that you could use it as a tool to identify places that need attention. But I don't know, I think I'm just guessing that could be what the objective of the study is, like, I don't know. And then maybe I'll go to Eric. Eric, I really liked your study as well, just as I did Maria's. And my question is, are you also planning to do some economic analysis, or do you have some economists as part of the project? And then the second question is, how long has these experiments been running?
So the first question, I'm gonna do like nitrogen budget, and then at the end I'm gonna try to put a dollar value on the nitrogen, but that's all. I have someone that's gonna be taking account of the ecosystem services as well in the trial, and then I first started the trial in 2017, and it's still going on. So next year, in all the treatments that we have in there, we're gonna plant peanuts. So, and then we're gonna see the effect of the long-term rotation. It's not long-term rotation. It will be like 3 years on the peanuts, and then I'm gonna repeat again. But it's not gonna be me anymore. It's gonna be another student.
Okay. No, because
for 4 years already, so it's
okay. So, no, the question was really because I mean, if you're thinking of, if you're also looking at carbon changes, you probably need a longer time to see some of these changes.
Yes.
But you could get some indicators, right? If you're looking at, like I saw you were looking at particulate organic matter as well, right?
Yes, I am, just because we can see like the changes more quickly. So that's why I'm looking to this organic matter, to the particulate organic matter. I'm also doing the carbon fractionation using the sieves from 0 to 15 centimeters. I use like 4 sieves, and that's another variable that I'm gonna be measuring there.
Yeah.
Yes, but yes, the trials is still gonna be running for some years.
Yeah, I think also for you, I think that will be one question you will get, the economics part. Definitely in your defense, someone is going to ask you about that. So maybe just make sure that you also have those numbers in place in terms of, will the farmer adopt, and what would it take for a farmer to adopt? Do they need government support, or do they need support from anyone? But the numbers, in the end, there's a lot of technology, but sometimes everything just falls apart on numbers, right?
For example, for the first chapter of my thesis, and the one that I'm talking about, the litter decomposition, I'm putting everything on a nitrogen basis, how much nitrogen is going back to the system. And I forgot to mention, but it was in the pictures. I'm evaluating the nitrogen return from the above ground portion of the plant, and also from the roots. So it's not only the above ground part that's going to be contributing to the nitrogen and nutrient cycling. So we are also accounting for the roots, which is really important. But yes, at the end, I'll try to put some conclusions on the dollar value, but mostly I'm focusing the nitrogen amount.
Thank you.
Which is gonna be, I mean, if I put a value on the nitrogen amount that you can save, and also on the cotton yield, it's easy to do that, but it gets more complicated when we try to put the animal data into account, right?
Yeah.
Because we are just mimicking the grazing there. We don't have the animals there for the whole month, for example. So it gets more difficult to put the animal value on that, but we can do estimates because we have some similar trials. I mean, trials that the animals are grazing the same type of forage. But yes, I'll do add some content. Thank you for the question.
And then I have a question for Ricardo. Just in terms of nitrogen application in Brazil, is it a common practice? And if it is, are you taking that into account in your experiments? If it is not, then probably the focus would then have to be just urine and dung, right? I don't know. Is it a common practice for farmers to apply nitrogen in pastures? And the rest for Ricardo. Okay, so maybe if Ricardo, maybe he could answer later. But then the final question I have is for Anthony. And Anthony, the question is related to.
I see Ricardo's
carbon data you showed. Do you have some? Okay, sorry. Okay.
Are you there, Ricardo? Ricardo, please feel free to respond to Ngoni's question.
Hi. Can you repeat, please, the question?
Okay, so the question was, in your experiment, you're looking at bracharia, right? And the question is really, in terms of nitrogen application, is it a common practice for farmers in Brazil to apply fertilizer in pastures? And if it is, are you applying the rates that are you going to apply the rates that they use? Or if it is not, are you then going just to focus on urine?
Thanks for your question. fertilizer with the nitrogen in pasture is not very common. Is some pasture is this use nitrogen, but is and can I say extensive system, extensive system, but in common areas, it's not common applicate the nitrogen because and I use the urine in my experiment because this some producers use the nitrogen and the others is not used. I don't know if I respond to your question.
Okay, so if you're not using fertilizer in your experiment. I thought on one of the experiments you said nitrogen application, but maybe I got it wrong. But yeah, I think that's fine. And then the next question will be to Anthony. And Anthony, you showed us some carbon values, but my question, you showed that the carbon was going up and down as you go down with depth. But do you have any uncertainties in terms of errors for these numbers? So will they be significantly different, or just probably not something to worry about?
Yeah. Sorry, I see that Anthony's dropped off the phone call, but that would have been a good question to know the answer for.
Okay. Okay. So otherwise, I'm okay. So I think I'll just follow this.
Perfect. How about if any of the other students, does anyone have any questions that they wish to ask? Feel free to raise your hand or unmute yourself and ask a question. We currently have Maria, Eric, Ricardo on the call. So Nelly, please go ahead.
Hello.
Hi Nelly, we can hear you.
Okay, good. Thank you for all the presenters. Actually, my question goes to George, specifically to his methodology. There are several methods to evaluate soil microbial diversity. So I would like to know why he has chosen chlorophall irrigation in spite of some others accurate and more efficient techniques like a PLFA, for instance. That's my question.
Unfortunately, Nelly, I don't believe we have George on the call. And we just had a little bit of a bad connection with your question there as well. But if you email that question through, we can pass that on and make sure it goes to him.
Okay, I'll type my question then.
Sounds good. Does anyone else have any questions for either Maria or Ricardo or Eric? Maybe Anthony might join us again. No questions. All right. With that, Ngoni, did you have any final comments to make?
No, I think I really enjoyed listening to the presentations and seeing the direction the CLIFF-GRADS students are taking. And I'm really looking forward to seeing the results and the outputs. I don't know if there's going to be some webinar later, Hazel, where people present the results of their work or something. It's interesting now. It's kind of like an appetizer for us, right? But it should be interesting to see what comes out of it and what are the outcomes and the outputs of this, and then maybe we could also discuss what are the application and what does it mean to the farmers and what does it mean for the climate and how can we use the results to inform policies and to inform management practices that farmers are taking. So I think it will be interesting later to see what comes out of it.
Yeah, absolutely. And hopefully there will be some opportunity in the future to discuss the results of all of the research, and hopefully that discussion might even be able to be held across multiple different rounds, because obviously these are just the round 3 students, and once the round 4 students have completed their research and we obviously have more of the older alumni from round 1 and round 2, it would be great to get everyone together to discuss. So yeah, thank you once again, Ngoni. It's been lovely to have you, and lovely to hear the bird song in the background from you as well.
Okay, thank you.
Yeah, thank you to everyone for joining us. Thanks to all of our student presenters, and yeah, I hope you all have either a lovely evening or a lovely day. So, bye everyone.
Thank you. Bye-bye.
Bye, thanks.